We propose a general model for stochastic genetic oscillations. Our model consists of a time delay nonlinear differential equation with multiplicative noise. Our main variable in our model is W, which is the concentration of the protein inside the cell associated with the genetic oscillator, and it has a linear decay rate, a production rate, which is gated by a time delay negative feedback, and it has multiplicative noise. In our theory, the production rate can be stationary and constant, or it can be time-dependent and noisy. We have shown in our paper that this model can be characterized by 4 stochastic variables. One of the stochastic variables is an approximation for the statistics of the maxima that we see in each oscillation cycle. The other one consists of an approximation of the minima that we see in every oscillation cycle, as well as the time that it takes to fall down from maxima to minima, and the times that it takes to go from minima to maxima. We have used these stochastic variables that also can be extracted very easily from experimental data, and we have compared with 2 different cases.
In the first case, we studied the dynamics of the circadian clock, which regulates the timing of different physiological processes around the day. And in the second case, we studied the segmentation clock, which regulates the sequential formation of tissue blocks, called somites, in early embryo development. In the case of the circadian clock, we analyzed data from single mice fibroblasts expressing a luciferase marker tagging the protein PER2. In panel A, we can see that the oscillations of a single cell are self-sustained. Our model reproduces this case by having a constant production rate. In the segmentation clock, cells make collectively a traveling wave in the prismatic mesoderm. We have extracted the cells from the prismatic mesoderm to see their behavior in isolation. In panel D, we can see a time trace obtained from a single cell. We can see here that the oscillations are non-stationary, with an amplitude that initially increases and then decays. Our model reproduces this case by having a production rate that initially increases as a noisy ramp and then it decays. Our model reproduces well the statistics of extrema and timing. Even though both systems operate within different biological contexts, they show generic features as well as differences. Our work shows that genetic oscillators with diverse biological contexts can be understood in a common framework based on a delayed negative feedback system and regulator dynamics.